From Microbe TV, this is TWiP, This Week in Parasitism, episode 182, recorded on April 9th, 2020. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hello there, Vincent.
And from a remote location, Daniel Griffin.
Hello, everyone.
Hey, Daniel.
Just like the old days, huh?
I tell you. the tried and true trio.
If you like what we do here on Microbe TV, consider supporting us, microbe.tv/contribute. Well, Daniel, it's good to see you. You're in your home. You're actually having some time off.
This is true. Yeah, this is where I record, a lot of those interviews that people may have seen on like Fox News.
Oh yeah, I watch that every day. I'm glued to the set.
I've got to put like a different backdrop. I always have to like clean up because my kids make a mess here. So always like before I interview, I'm like throwing stuff, covering it with a blanket.
That's their job.
I'm like, there's glue. Why is there a bottle of glue?
Really?
I guess during the pandemic, there's lots of glue and projects going on.
Absolutely.
If you want to hear COVID-19 updates from Daniel, tune into TWiV. Tomorrow's Friday. We'll be recording one, right, Daniel?
Yes. Looking forward to it.
From a parking lot, a hospital parking lot.
That is the safest zone. I go out into the Tesla and I turn on the biosafety, button there.
The UV lights come on and sterilize the air coming in through the filter.
Yes.
Today is TWiP, This Week in Parasitism, and let's start with revisiting our case from TWiP 181. Daniel.
All right. So now we are case from TWiP 181. So for those of you tuning back in, and those of you tuning in for the first time, this was a case of a volunteer from a previous case. This poor individual, right? Not just one thing. Occam was, I guess he was with only one thing at a time, but so maybe this person had two things at a time.
It's a double-edged razor.
Exactly. So this volunteer had diarrhea before. We had addressed this issue, but now they had a second issue. So here we are in eastern Uganda. We're up on the really right next to Kenya, right on the border, under the shadows of the mountain. And this individual is volunteering at the Baduda District Health Center for FIMRIC, Foundation International Medical Relief of Children. And what she started to notice were these skin lesions. And she would notice these when she would wake up in the morning. And there would be a series of about 1 centimeter or smaller red raised lesions. They would be somewhat in a line. There might be 3, there might be 4, and they were mostly on the torso. She would go to sleep without these. She would wake up with them. The areas were a little bit swollen or quite itchy. And as I mentioned, every morning, not every morning, but some mornings she would wake up and there would be new ones. And so she asked Nick, the manager of the Uganda site, what could this possibly be? And he said, well, welcome to Uganda. And so she was not happy with that response, that this was just, part of the experience. So she moves out of the room and she leaves everything behind. And she's now basically sleeping on top of a plastic covered mattress in the other room. And she notices that when she wakes up, there are no new lesions. And so she has a sort of a mixed response, like, okay, so, the problem is somewhat solved, but I can't really just sleep on this plastic covered mattress in this other room. What's going on? When can I move back into my room? What are my options here?
Okay. And, despite the turmoil going around in the world, we do have quite a few guesses. So that's great, listeners. I guess you're all sitting at home. This is what you're doing, right? All right, our first is from John, who writes, Hi, TWiP hosts. This is my first time writing in after listening to many of the episodes since coming across the podcast in my second year of undergrad studies, of which I'm now currently in my first year of MSc in microbiology.
Nice.
I believe this risk of being helpful has led to the volunteer becoming the nourishing blood meal for one of the 5 nymphal stages of Cimex hemipterus, commonly known as the tropical bedbug. The series of lesions that appeared to be the paired bite marks, breakfast, lunch, and dinner, which are supposedly characteristic of Cimex species compared to single bites from other insects. Although this cannot be the sole reason for diagnosis, and actual identification of the insect is needed for confirmatory diagnosis. Moving from the bed of origin, deep cleaning of their clothes, especially with a hot dryer, and treating the room with insecticides such as pyrethroid and neonicotinoid would hopefully eradicate the pests. P.S. Coming from Cape Town, South Africa, where TB and HIV are endemic, leaves me wondering how devastating the effect of COVID-19 may be on these vulnerable groups of people.
Right.
Our government has recently taken action to slow the spread, as this would be vital to make sure there are beds available for severe cases that may arise from those that may have HIV, TB, or both. Have there been any reports of individuals with both TB and COVID-19, and what are their outcomes? Wishing you all the best, Josh.
Well, there's a lot in there. It's a pretty sophisticated, right? Making these subspecies distinction here between Hemipterus and Lectularius. So.
That's right.
But yeah, actually, as we get near the end, maybe we'll talk a little bit about the impact of COVID-19 on some of these global health sites.
Terrific.
Dickson.
James writes, linear bites, the differential diagnosis is short. One trip my wife and I made to Seattle to visit the grandkids, and my granddaughter too, and my daughter too. We stayed in a nice hotel and also in a really nice hotel in Vancouver. We noticed linear bites that itched. The red marks were a lot less than a centimeter, more like 3 millimeters, but it was the first time I'd ever been exposed to bedbugs. Cimex lectularis. Ick. We washed and dried everything, and thank God did not bring them home to Denver with us. But then my brother called and said he was being evicted from his apartment due to the bed bugs. I was frankly more worried about them getting a ride from him to my elderly parents' condo in a big building. What a disaster that could have been. As I recall, it was summer, so we packed up his stuff and put it all in an unventilated storage unit for a while and baked the little buggers. We looked up and found a whole variety of homebrew bedbug traps, often based on carbon dioxide emission with dry ice, vinegar / soda, and even sugar and yeast. Well, I'm a mycologist, so I should say Saccharomyces cerevisiae, right? Drawing the bugs into various sticky places. These dudes are found in Uganda. Google showed them in all kinds of hotels there. In my active pathologist days, the infection control folks knew that there was this weird guy in the lab who liked to identify weirdness, and often they appeared with usually a urine cup, held carefully in gloved hands, usually coming straight from the ED. Usually it was either lice or bed bugs. I had to instruct the pathology assistants and fellow pathologists not to embed the bugs for histology. A thin 2D slide through an insect is so unhelpful. A hand lens, on the other hand, reminds me of the old pathology saying, high power lens, low power mind. I'd never heard that one before. I like that. I read that the eggs hatch in 6 to 10 days, and both nymphs and adults need blood meals. Now, this is a huge bummer. Nymphs and adults can live without a blood meal for months to maybe even a year. Talk about intermittent fasting and low-carb diet. There are, on the UMN extension site, all kinds of delightful pictures of bedding with bloody bedbug poop stains. I suppose it would be prudent when traveling to look at the bedding, but does anybody really do that? Thanks for the memories, James. He's an MD and a PhD associate professor. I love it. And that was a great take on that. Thank you.
My wife always tells me to look at the bed when I travel.
They're under the bed and they're in the cracks in the walls. They're not in the bed. They're in the cracks in the walls, trust me.
Well, James said there are bedbug poop stains on the sheets.
Yeah, no, that's when they bite. That's the poop. Then they go back to the cracks on the walls afterwards. It's true.
Daniel.
Andrew writes, Kia ora from Pongaroa. No book one yet, but I see sending a picture had a magical effect last time. See below. The weather is dry and windy, 19C. The drought continues. Pongaroa has no cases of COVID-19 yet, with only 28 cases recorded so far in the country, all infected overseas or close family, and our international borders are now closed to all but citizens and permanent residents. It might be a while before Pongaroa will see any cases, but we are routinely prepared to survive alone for at least 2 weeks due to the earthquake risk. It will be interesting to see how many cases there are when the next TWiP is produced. My guess for this month's case is Cymex hemipterus, the tropical bedbug. I have had experience with their temperate cousins, Cymex lenticularius, and recognize the line formation of the swollen lesions the young lady had. The main difference between the two types is morphology, as far as I can determine. The tropical variety have necks, or not that they are just as execrable. They are tenacious creatures, and unless the bedding is washed in very hot water and the room and furniture is carefully inspected with a magnifying lens and every adult, nymph, and egg removed, they would need to wait at least 4 months, but probably longer due to the higher temperatures and humidity one would expect in Uganda. Perhaps 18 months before the last egg has perished. Now to my picture. I was made aware that 2 young PhD candidates who are researching in microbiology would like to share their love of science with the people of Pongaroa. As it is the birthplace of Nobel Prize winner Maurice Wilkins, there was a definite hook to microbiology. We organized 2 events, a talk for the adults and an experiment at the local primary school. While the adult event was a success, the school kids had a blast extracting DNA from kiwi fruit. The young PhD students shammed it up by wearing lab coats and safety glasses, and the teachers have prepared the children with age-appropriate explanations of what scientists do and what DNA is. The little kids mashed up the kiwi fruit, and the older ones mixed the buffer solution and poured the methanol into the beakers. They all watched the DNA precipitate out with wonder and delight. We are sure that some of those children will be inspired to pursue science as a career. And who knows, Pongaroa might get its 2nd Nobel Prize winner in due course. We could not obtain ethanol for the experiment, as most online descriptions recommend, but the purple color of the methanol used made for a rather fetching combination with the green of the kiwi fruit and the white of the strands of DNA. And he's sent a picture. If any listeners are interested, maybe they have bored kids in quarantine or lockdown. The experiment is detailed at the Naked Scientist website. And he gives a link there. Matewa, see you later, from Andrew. And we've got a wonderful picture of this beaker.
It looks like a zombie. I mean, I would almost be tempted to drink it, except for the methanol.
Keep it away from Dickson.
Keep it away from Dickson. That's the idea.
I have to say, as a kid activity, people love DNA extractions.
This is true.
We do it with bananas. I've done it with bananas a lot. Even teenagers, so.
Mm-hmm. Nice. Very pretty. All right, David writes, Dear TWiP hosts, I believe the parasite-plagued patient from Uganda is suffering from the bites of bedbugs, hematophagus insects in the genus Cymex, which have a penchant for human blood. The 3-4 raised lesions in a line on the torso were a dead giveaway, and seeing the lesions disappear after the bedding was left behind is another insightful clue. I remember Dickson once saying 3 lesions in a line of this sort represent breakfast, lunch, and dinner.
Did I actually say that?
Apparently.
Okay, I'll take credit for it.
And maybe the 4th could be dessert. Unfortunately, bed bugs can be rather difficult to remove and require pesticidal usage and mechanical controls, removing the bugs with a vacuum, heating up the mattresses. Depending on where the eggs of this insect were laid in the room, it can take quite a long time to fully eradicate the bed bugs. New York State, I guess, or New Stork. No, it's got to be New York.
New York State.
Integrated Pest Management claims it may take at least 3 weeks to get rid of bed bugs, but this information is coming from New York, not a resource-limited region such as Uganda, where some of these pesticides or amenities may be lacking. Thank you once again for the informative and entertaining podcast, and I hope you are all keeping healthy and safe. David P. P.S. I wanted to share with you and the listeners my first research article published in Biology Open since I joined Tufts Vet School as a PhD student. I cannot believe after starting to listen to TWiP 4 or 5 years ago that I would be a published author in the field of parasitology. Your podcasts have served me well in my time in and out of the lab. Thank you for being an inspiration to eager scientists and listeners alike. So he sends a link to his paper in Biology Open. Schistosome mansoni glyceraldehyde 3-phosphate dehydrogenase enhances formation of the blood clot lysis protein plasmin.
Look at that.
Nice.
Look at that.
Thank you.
That's great.
Good job.
Absolutely.
That's some solid science there. That's exciting.
I think David has won a book, so I'm going to take a number off of him there.
So
Dickson, you are next.
I am. Schuler or Schuyler?
Schuyler,
Schuyler writes, Dear Twipsters, I hope you are all staying well and washing your hands like someone scrubbing in for surgery. I used to listen to TWiP when traveling home during freshman year of college, 2013, and recently started listening to podcasts again, including the wonderful TWiP. I like to listen to your podcast, especially when going for walks. It is a pleasant 50 degrees F here in Cambridge, Massachusetts, although it's a bit overcast. I must say, I have 3 motives for writing in this week. First, I just listened to TWiP 177, where you discuss the scabies is specific to humans and could therefore theoretically be eradicated. As someone who considered veterinary medicine before deciding to pursue human medicine instead, I wanted to let you know that the sarcoptic mange, aka scabies, affects dogs as well as humans and is not specific to humans alone. The second form of mange, Demodex, is specific to dogs. Poor dogs. Second, I would absolutely love a book, as you have not, as I do not yet have one, and I would love to bring it home with me when I start medical school at Georgetown this summer. Third, I finally listened to a recent episode instead of an old one, and guessing seemed like fun. My initial suspects for Case 181 are bedbugs and kissing bugs. If it were not some sort of internal parasite, the lesions wouldn't stop after changing rooms unless the patient happened to move at the time the disease resolved itself. Well, the CDC website says kissing bugs that transmit Chagas disease only live in the Americas, so that's out. So I thought. The International Association for Medical Assistance to Travelers still warns against kissing bugs on their Uganda page, so maybe another variant that doesn't carry Chagas lives in Uganda? Regardless, I think bed bugs are more likely, though. I find it a bit odd she got so few bites per night. Perhaps the infestation was not that bad yet. The line of bites is apparently a sign of bed bugs. Reference 3 and 4. Wikipedia refers to these as breakfast, lunch, and dinner. See, I think that was Wikipedia. I don't take credit for it. And the IAMAT warns of bedbugs in Uganda as well. I'm not sure I would return to the room or even the same building. My understanding is that eradication of a bedbug infestation is a nasty thing, requiring extensive cleaning, heat treatment, and multiple washes of all affected cloth. Of course, if you starve the bugs, they'll eventually die. According to the University of California Agriculture and Natural Resources, bedbugs can live 20 to 400 days without feeding, depending on the age of the bug. Adults can go 400 days without feeding, so I will say no one should use that room until at least 400 days have passed. That's very smart. But the woman should dispose of or treat any of her belongings that could be carrying bedbug eggs or nymphs from the old room to the new one. Wishing you all the best. Oh, it's pronounced Schuyler. I can see that. Wow. Thank you, Schuyler.
You put it right at the bottom there.
Thank you, Schuyler, for all of those bon mots. Then he lists all these references down at the bottom of his.
Yeah, so should we comment about this scabies thing? Should I jump in on that? So,
Go for it.
Yeah, so what she says is actually interesting. So there is a canine scabies known as mange, and those mites actually can get onto humans and cause us itching and unpleasantry, but they can't multiply, and so they're only going to live for a limited period of time. And the same is this issue with the human scabies. They can actually get onto dogs and other pets, but again, they can't survive for extended periods or carry out their life cycles. So, but if you think about the sort of phenomenon, right? If they're, most of the time as we're describing, they're getting onto a person, you don't care if then they go off and bite a dog and have their life cycle. So yeah, there is this interesting relationship here. The scabies need humans to survive and undergo their life cycle. They can, I'll say, harass and torment other creatures, but each scabies, in my understanding, requires a specific host for its replication.
Yeah, I think
cycle.
that's what I've heard too, by the way, Daniel. So you're up, Vincent.
400 days. 400 days is a long time.
It is, isn't it? But not as long as some ticks. Some ticks can go years without feeding. So that's
amazing.
I know. It's really depressing when you think about it. I can't go 2 days without eating.
Okay.
Daniel.
So, Western U, is it me?
Yes,
Am I up
it is.
next?
Yeah.
So the Western U Global Health Track students, Dana, Stephen, and Chris write, we believe the most likely diagnosis to be bed bugs. Bed bugs normally bite 3 to 4 times in succession over a few minutes to engorge themselves. They would appear symptomatically as lesions in a line like the patient is experiencing. Also, the fact that the lesions stopped after they left the room and bedding behind suggests an infestation in the bedding. However, identification of the insect is required to confirm the diagnosis. As for moving back into the room, it'd be best to have the bedding washed and put through a hot dryer to kill the bed bugs. Vacuuming can have a significant impact on reducing bed bugs, but considering the floors are concrete, that probably wouldn't help. Maybe they could sweep the floor every day. The bed bugs could be infested in the mattresses as well, so possibly these would need to be replaced. Alternatively, the room could be treated with products containing pyrethroid and or neonicotinoid. According to your book, hosts can become desensitized to bed bug bites over regular exposure. So possibly the volunteers could be told to wait it out. But that would probably be very annoying to hear as a patient. Maybe the desensitization bit is why the manager was so casual about the whole situation, since he has possibly been there long enough to reach that point. Thanks for all you do. We really appreciate the COVID-19 daily updates on your website. Sincerely, Western U Global Health Track students, Dana, Stephen, and Chris.
Cool.
So that update you can find at parasiteswithoutborders.com. There's a COVID-19 button on the front page. You can get daily updates, or almost daily. Right, Dickson?
You betcha.
Evan writes, I hope you're all doing well amidst the coronavirus pandemic. Here in Virginia, we are trying our best to stay at home, and all medical school classes have moved online. With my newfound free time, I've been able to catch up on my podcast. I hope I am not too late in entering a guess for the case from 181. Here we have a medical volunteer in Uganda presenting with red lesions clustered on the torso upon waking in the morning. Lesions are absent upon moving to a new room to sleep. I believe this is a case of bed bugs, as the rashes match the classic description, and there are multiple, which rules out other common bed bugs stings. Ideally, the bedding should be washed and room cleaned to get rid of the offending buggers. Hopefully this is correct. If not, I'll be sure to check out PD7 soon. Wish you all the best. Look forward to more TWiP. Wow.
Right. Peter writes, the description of lesions appearing overnight that appear to be associated with a particular room and lodging suggests to me that the culprit here is the same as the one discussed in TWiP's 100 and 101, the bedbugs. The bedbug Cymex lectularis or Cymex hemipterus. The description of 3 or 4 raised red lesions in a line corresponds to the apocryphal Rule of 3 Bites, Parasitic Disease, 7th edition, page 470. Treatment of the room is with insecticides, pyrethroids, or with neonicotinoid-based product. Vacuuming, washing of all bedding, covers, etc., and heat drying, and ensuring that cracks and other hiding places are covered up when possible. Encasing of mattresses in plastic covers is also recommended, referred to by the U.S. EPA. Sounds like this last advice could leave you with a crinkly night's sleep, though. The room can be reoccupied after treatment, that is, in about a day's time, reference Michigan State University. I do not know how much the above advice needs to be adapted to the conditions of warmer climes. And then he gives some references, and he keeps going, and that's... should I keep going? Because I think it's the same.
You gotta get to the poem.
It's same Peter. Okay, so Peter. He signs off by saying, locked down in a Cape Town that is turning to autumn and colder evenings. P.S. Professor Despommier often mentions the story of Mao's army, the planned invasion of Formosa and schistosomiasis. I do not know if he is aware that Mao Zedong wrote a poem about schistosomiasis. Farewell to the god of plague. Mao writes, when I read in the Renmin Ribao of June 30th, 1958, that schistosomiasis had been wiped out in Yongkang County, thoughts thronged in my mind, and I could not sleep. In the warm morning breeze the next day, as sunlight falls on my window, I look towards the distant southern sky, and in my happiness pen the following lines. So many blue streams and green hills, but to what avail? This tiny worm defied even the greatest physicians. Hundreds of villages choked with weeds, men wasted away, thousands of homes deserted, spirits chanted mournfully, merely sitting there. Daily I travel 80,000 miles a day, surveying the sky. I see a myriad of Milky Ways from afar. Should the herd boy star ask tidings of the god of plague, I'd reply, his joy is gone as our sorrows. The spring wind blows amid profuse willow wands, 600 million in this land, all like saintly sires. Crimson rain swirls in waves, green mountains turn to bridges as our wish. Gleaming pickaxes fall on all of China, from the 5 ridges of the Yellow to the low rivers. Mighty arms pound the rock to earth. We ask the god of plague, where are you bound? Paper boats lit by candles illuminate the sky, taking the demon god away. The herd boy star, otherwise translated as the cowherd, is a reference both to Altair, that's a star, and to the story of the cowherd and the weaver girl. The poem feels quite fitting at these times. Source of the poem, then he gives a good source of the poem. That's great.
Do you know about this poem, Dickson?
Nope, I never heard it before in my whole life. Maybe we'll put it in this 8th edition of Parasitic Diseases under the Schistosome chapter.
Makes sense.
Could happen.
Yeah. Daniel.
All right. I like that, actually.
Good.
Cecilia writes, Dear doctors, your volunteer in Uganda was probably sleeping in a room that was infested with bed bugs. Cymex lectularius and C. hemipterus. Hide in the seams of bedding, crevices of flooring and furniture, and behind peeling wallpaper. The bed bugs come out at night to feed by biting the victim for a blood meal. This nocturnal feeding results in more than one bite, usually 3 to 4. This can be differentiated because from the bites caused by bed bugs and mosquitoes or fleas. The infested room needs to be treated by washing the bedding and any clothes that may be stored in the room. In addition, the mattress and surrounding area should be inspected for evidence of bed bug infestation by looking for the bugs and feces in the seams of the mattresses and any cracks and crevices. Thanks again for the podcast and the interesting case studies. Sincerely, Cecilia from St. Petersburg, Florida.
Ben writes, to the TWiP-tastic trio, this study sounds like a classic case of bed bugs. Getting rid of bed bugs can be difficult, but can be achieved through use of insecticides, although resistance is increasing, or more mechanical approaches like steaming of beds, etc. Though I thought I would add a paper suggestion. This is a bioRxiv preprint. The authors investigate 2 proteins and how they're involved in microtubule assembly and anchoring to the conoid of Toxoplasma gondii. In particular, the paper has some absolutely beautiful microscopy with the first published example of expansion microscopy in apicomplexan parasites. Since the last TWiP episode, my first author paper was published where we look at a protein involved in malaria parasite invasion. We did quite a lot of super resolution microscopy in that paper, and I'm hoping to use expansion microscopy on malaria parasites in the near future. So I was very excited to see this.
Cool.
Keep up the fantastic work, Ben.
Very cool.
And Ben is in Adelaide.
Adelaide. That's right.
Dickson. Guess what? You got
Kevin.
You got the jackpot, Dickson.
I got it. Quotes. There are other health impact. Kevin writes, quotes, there are other health impacts of bed bugs. The challenge and costs of pest control often lead people to desperate and dangerous acts. Quotes from Doggett et al. Another quote, that nauseous, venomous insect from Southall, 1730, a treatise of bugs, 2 G's, by the way. The volunteer nurses from TWiP 180 recovering from severe cyclospora diarrhea now have experienced another degrading visitation, nocturnal assassins. I won't beat about the bush, but habit forces me to make a brief differential diagnosis. Patients often present with a lesion that they insist is a spider bite. Why is this? Anyway, our volunteers do not seem to have scabies, bites of the tomb bove, floor maggot, mosquito bites, lice, or cutaneous larval migraines. Here be Cymex, bed bugs, either Cymex lecularis or the tropical bed bug, Cymex hemipterus. One old references mention slang terms like mahogany flats and redcoats, but one wonders how often these terms were used. Despite the now decades-long global resurgence of bedbugs, I am aware of no common slang expressions. Massive amounts of treasure have been expended in eradication efforts, but the bugs endure. Regarding our African location, the Nigerian reference below states that only 16% of inspected dwellings were without bedbug infestations. I will list some essential facts that may assuage a few of the more irrational fears that bug victims nourish. The bullet number 1, bedbugs carry no known human disease despite laundry lists of his suspects that have appeared through the years. And that Brooke Blumberg championed that research, by the way, I must say. Second bullet, bedbugs cannot fly, hop, jump, or perform other acrobatics, such as fleas can. I just added that myself. The third bullet, they can live for over 1 year without feeding, but normally live 4 to 5 months under normal household conditions. Another bullet, bedbugs do not live upon the host. They are hit and run raiders. I have seen the molted exoskeletons on people, however. Another bullet, some people are seemingly unaffected by bedbug bites where others can have exuberant or even bullous lesions. Another bullet, the old saw that bedbug bites occur in clusters of 3, the so-called breakfast, lunch, and dinner pattern, has been largely debunked as oversimplification. Doggett, however, the Australian Cymex guru, presents good evidence of linear and clustered biting patterns. However, and his 2012 review shows clear photographic evidence for this. I would like to further calumnize the bedbug by first mentioning its legendary disagreeable odor, described in detail by most authors. I regret to say that I have personally been in rooms that were heavily infested and never noticed this buggy odor. Must get my olfactories checked. Another despicable characteristic of these creatures is their practice of traumatic, also known as hypodermic, insemination, despite the female's possession of perfectly formed genitalia. The male bug mates by directly piercing his partner's abdomen with his foul aedegus. I will finalize this gerrymade by repeating a finding from 1976 where the orthobunyavirus, king koi virus, was recovered from bat bugs in Thailand. Vigilance is required regarding the bat bug matrix and the theoretical potential for some kind of spillover of bat viruses to bat bugs and hence other mammals. What do our volunteers do now? Professional extermination services are probably hard to find in Budaba. If insecticide spray is not available, cracks, crevices, etc. should be inspected and manually cleaned. The bedding should be completely inspected and, if possible, encapsulated in bug-proof mattress covers. Placing the mattress in direct sunlight for several hours may have some effect, but sustained temperature of 150 degrees Fahrenheit are required to kill the bugs. Bed furniture, springs, etc. must be inspected and cleaned. Once the bed and mattress is determined to be bug-free, bed isolation can be performed. Pitfall or sticky traps on each of the 4 legs of the bed, taking special care now that no sheets or blankets are in contact with the floor. Pitfall traps can be filled with a borax solution or diatomaceous earth. This is not a permanent solution to the problem, but may help at night. The WHO states that pyrethrin impregnated mosquito nets may kill bedbugs. They also note that DEET can mitigate biting, but the deterrent effects will not last through the night without reapplication. As early as 1730, simple, unadorned, and easily cleaned bed furniture has been recommended to deny arborage of the pests. An elderly patient I once knew had an extensive infestation in her bedroom, especially involving her ornate headboard. When advised to dispose of this furniture, she offered the strictly reply, "Not my French provincial," thanking twixt humans, super spreaders of science. We're super spreaders of science. I love it.
I like that.
Now, how much of this other stuff do you want me to read?
No, you don't have to read. I would just like to point out that there are extensive...
It's a wonderful treatise.
There are extensive references, and I want to point out a terminal curiosity. Bruce Frederick Cummings In 1919, wrote the very famous diary Journal of a Disappointed Man, which, among many other things, is one of the best-known accounts of multiple sclerosis ever published. Of interest to us here is his 1917 publication, The Bedbug, Its Habits and Life History and How to Deal with It.
Right. I must say also this idea about putting some tape or some trap on the legs of the bed. I once heard a story. I don't know how apocryphal it is, but I presume a little bit. Someone decided to put the legs of his or her bed in containers that were filled with cooking oil.
Yeah.
So that the bed bugs, when they tried to get into the bed, of course, when they crawled out of the walls, they had to climb over the edge of this thing and they fell in and they drowned right in the oil or they got stuck in it. But that worked for the first night. The second night, the person turned the lights on halfway into their sleep because they were awakened by these itching and biting things and all that. And they looked up and they found a trail of bugs going from the wall to the ceiling over the bed, and they were dropping off of the ceiling down onto this guy's bed. Mm. Now, if they're that clever, then I think that they should be given a PhD or something like that.
So Kevin has a page from a treatise on bugs, which is free on Google Books and archive.org, and he has drawings of nits,
they're great.
And bed bugs of different ages. You can see how big they get. So that's very nice. And then he talks about the annals of the hair of the dog that bit ya. Review discusses the use of bed bugs in traditional medicine. An example is treatment of ringworm in India with Simex crushed in the herb holy basil.
Ugh.
Other Indian bedbug containing remedies were used for the treatment of epilepsy, piles, alopecia, urinary disorders, and snake bites.
Good heavens.
Greek army surgeons around 50 AD claimed that bed bugs could neutralize snake venom. And he sends a picture of a typical bed bug trap, like you're just talking about, Dickson.
Yeah,
where you put it around the bed leg, and the bugs fall into it. I guess you could put your oil in there, right, Dickson?
You could do that, but they're so clever that they'll just climb up over and then drop off the ceiling on you.
Wow! Thank you, everyone, for your guesses. This was lovely, and please, Daniel, what else do you have to tell us?
Well, I think now we have the last two guesses.
We do.
We seem to have a consensus, so I guess we'll
This is
see
a pretty no-brainer, I would
think.
say. Yeah, go ahead, Vincent. You go first.
Dickson, when we did this, I think you and I were still in the office at Columbia, right?
We were.
And so when you were describing these, Dickson just did a thumbs up. And we both felt that we had bed bugs at the time, didn't we?
No, we did. We actually did. Yeah, I think that this is a no-brainer. Basically, that if somebody guessed something else, then they weren't aware of the world we live in.
We had one possibility of a Chagas, but that wasn't.
Nah, they didn't choose it in the end. They chose the bed bug. Yeah. So everybody got it.
No, I think that, hopefully this for most of our listeners was an easy one, shall we say?
Exactly.
One where they could feel good about getting it right.
Exactly.
And but the emailers brought up some interesting points, I think.
They did.
So one of them was, and I know in our latest revision of parasitic diseases, are we up to the 7th edition now?
We are.
We did talk about, though breakfast, lunch, and dinner is easy to remember. Often these could be 2 bites, 4 bites, so it's not always 3.
Right.
It's often 3 or 4. So that's interesting. They are often linear, but I do think the breakfast, lunch, and dinner is a nice way of remembering. And I think I have coined the and dessert in the past. So
Griffin gets a star after his name.
Exactly. So we'll add that. But yeah, I think that there are a lot of features that people picked up on it. And a big one was, you're in an area where it is 84% of the dwellings have them. So sort of who doesn't have bed bugs?
Exactly.
Nick and I were not bothered by them because I grew up in New York City, so
right.
My body doesn't care if it gets bit by bed bugs because they're quite endemic.
Yeah,
you don't have to go to Uganda to get bed bugs, as a lot of people visiting New York have experienced. so I just basically let this individual know, like, you just have to stay out of the room for just a period of time, 400 days.
This is true.
Which she seemed to think was excessive. No. So, one of the challenges, as people brought up, was what do you have access to in Uganda? There's no dryer within any distance that, one can easily travel in from Baduda. So you can't put everything in a hot dryer. That was an idea. There's not a lot of access to insecticides and things.
Thanks.
So, so what we did in this case, a couple things. The first thing this individual did is they went to the nearby town and bought fresh bedding that was in a plastic covered case and basically stayed in that other room. But then, like, what do you do with all her clothes that are still in the room?
That's right.
So these were put in these black garbage bags, which we then left out in the sun. And so basically it gets super hot and it's a way to hopefully kill things. And then they were washed, and I think they went back in a different bag again. So quite a process when you have limited resources to deal with this. But then she did not move back into that room.
I don't blame her. So my wife and I were on a vacation down in Costa Rica not too long ago, and we were actually traveling down a road trying to see a jaguar in a jaguar reserve, and they had this study zone, these buildings that were made out of cinder block, and they had tin roofs and primitive living conditions for the people that were tracking these jaguars. And we had to sleep there that night. And of course, we slept 2 nights there. The first night, everybody woke up and everybody was bitten, except me. I was the only one that was not bitten, and I was sleeping on a bed which had a cover on it. All right. But I didn't use the sheets. I just used the bed itself. I didn't sleep with sheets on. So the next night when we went to sleep, I recommended for everybody to sleep with the lights on, because the bed bugs will come out when you turn the lights off. And son of a gun, if that didn't work. The next morning when we woke up, nobody had reported any new bites that night, and everybody slept with the lights on. So I know it's inconvenient to sleep with lights on, but it might be one way in an emergency situation to deal with it. And if anybody's got more experience with that than what I just related, I'd love to hear whether your experience was different than what I experienced.
No, and actually I'll confirm, because that is an interest, because of course I like to see these. But having a light on at night not only suppresses the biting behavior of bed bugs, but
mosquitoes.
Also suppresses the biting behavior of, mosquitoes.
That's right.
Yeah. So that's one of those interesting things. If people are willing to do this, is you can bring one of those eye masks, right? Where you put an eye mask over your eyes,
sure.
so you can sleep. This is something I picked up when I used to fish up in Alaska in the summers.
There you go.
But you leave the lights on, and they'll actually protect you from both bed bugs and so have an impact on this photic suppression of the biting activity of Anopheles mosquitoes.
How about that?
Wow.
A fancy way of saying they don't bite when the light's turned on.
That's right.
Yeah, it's cool. If you can sleep in the dark, there you go.
Yeah.
Good deal.
All right, so let's give away a book.
Got it.
We had 9 guesses, and let me do a random number between 1 and 9, and that number, Dickson, will be number 9.
Number 9 is, let's see, they're all numbered?
They're all numbered.
Ben, right? Ben was number 9 of the 9 without books so far.
Ben will be very happy.
Winner, Ben, send,
Excellent.
Darn, you're in Australia. It's going to take a long time to get to you.
He's a PhD candidate in a malaria lab too, so that should be great for him. Terrific.
But
it's good that it takes a certain amount of time, right? So that the virus that may or may not be on that book will have died during the transit process from the hot zone of New York.
They don't survive that.
Indeed. So, Ben, send us your address and phone. We need that for the overseas shipping. TWiP at microbe.tv. Good job. And you others, please keep trying. Ben has been trying a long time.
That's
right. You got it?
Right.
Time to do a paper.
You got it.
Yeah, I was going to, before we did the paper, I was going to, early on, someone asked about the impact of COVID.
Let's do that.
Yeah, so I thought I would, so the first thing I have to say, there was an immediate dramatic impact of COVID-19 on organizations like FIMRIC and floating doctors that I work with. A lot of these organizations receive their money from people donating who are going to potentially go to these sites. So Foundation International Relief of Children, basically in a period of 2 weeks, lost 95% of their income.
Wow.
Because you no longer had nursing students, volunteers going to these sites and donating to support the work. They also had an immediate impact on their people power because you could no longer be having people flying from the United States and other countries to a lot of these areas. And then you also had a lot of people leaving. Like, for instance, Nick left the site, the manager, to return to the U.S. during these times.
Wow.
So there was an immediate, really significant, dramatic impact on global health volunteerism throughout the world because of travel restrictions.
Sure.
Also, a really dramatic, significant impact financially on these organizations because no longer had the volunteer support and dollars coming in. And I think now what I'm quite worried about is going to be the impact directly of the virus, because here you have these communities. Now they have a lot less of the financial and medical support than they had before, and they already were, struggling.
Right.
And now you're going to have a virus sweeping into areas where there's limited hospital capacity, really not much in the way of ventilators and more advanced support. So I'm really quite worried about these areas of the world on all these different levels from COVID-19.
I haven't heard of any TB and COVID-19 patients, right? Have you?
So the TB is actually really interesting. And so one of the drugs I think that we've talked about for treating COVID-19 works on the immune system. And the type of immune system that protects us from tuberculosis is actually the proper type of immune response, is Th1 type response to protect us against the virus. And the virus is actually, if anything, gearing us towards this odd Th17 IL-6 driven response. So what we have seen, a couple things. One is, I don't know how much this is artifact, but people that received the BCG, so the tuberculosis vaccine at birth, if you look at those countries, and even in Europe where you have places where they do BCG or not, at least the early numbers were suggesting that maybe the TB vaccination was associated with lower mortality of COVID-19. And there certainly have been some tuberculosis and COVID-19 co-infected patients. And what it really is it's not necessarily that one increases your chance of the other, but they're both occurring in the same context. So you continue your tuberculosis drugs. And it's going to be interesting to see on large scales, do people with active tuberculosis under treatment do better or worse?
Interesting.
Yeah, but we're definitely seeing co-infections.
Wow.
All right.
Dear.
All right. Thank you. All right. On to our paper. Class pathogens. HIV-1 integrates widely throughout the genome of the human blood fluke Schistosome mansoni. First author Sutas Sutiprapa. Last author Paul Brindley. George Washington University. Mahidol University in Bangkok, Kon Kain University in Thailand, Wellcome Trust in the UK, Academia Sinica Taipei, Chinese Academy of Agricultural Sciences in China, and George Mason University, Virginia. Dickson, you picked this one.
I did, didn't I?
What caught your eye, Dickson?
Their conclusions, basically. If you skip down to the last part of the paper, they start to talk about lentiviruses as shuttle vectors for genetic manipulation of non-dividing cells. And I think that's a remarkable achievement, since they had nothing before that, and looked at this as a breakthrough in that technology, that you could now have a tool for studying the expression of various proteins, particularly the ones on the surface of the organism, which shields it from being attacked by the host immune system. The surface tegument of Schistosome mansoni turns over at a tremendous rate, but it also has mimic molecules to the host proteins, which sort of masks it from being attacked as a result. And if you could somehow alter those expressions using, like they said, lentiviruses that could carry the proper changed genes for those surface proteins into those areas of the parasite, you could then see whether or not it made a difference of the survival of the adult worms as they were being attacked by antibodies and that sort of thing. So I think it opens up a whole new field. And the other thing I picked it for was because I know that Daniel is intimately involved in HIV research, and you're a virologist and I'm not. And here's a wonderful crossover paper which sort of brings both of these areas together in a way which makes good sense. And it's the first time that HIV integration has been shown not to occur in a primate. This is not a primate, obviously, but because HIV-1 has some history of infection in primates, and Schistosome mansoni can infect primates. I'm just wondering whether or not this hasn't occurred in nature somewhere.
No, the answer is no.
You're sure about that?
Yeah.
You're sure about the Schistosome mansoni has never acquired HIV-1 integrase?
Can I jump in?
Yeah, sure. Jump.
When Dickson sent me this, it's a busy day in the hospital, sleep deprived as usual. I was like, do I have to worry about this too? Right. Isn't there enough going on?
Right.
But it's interesting because the technique that they talk about, this whole idea is they're using a lentiviral vector. So this is not wild type HIV that binds through particular co-receptors. This is you actually create in the lab these types of lentiviral vectors with what we call pseudo-typed envelopes. So this is actually what I do, sort of, I guess, on a regular basis when I'm hanging out in the lab at Columbia, is you take a couple plasmids and you create a new virus that doesn't exist in nature. And you have to do this in a BSL-2 and above level, because what you've created is a form of HIV that can infect any cell. You've basically taken away we'll say the restricted tropism, right? So normally HIV is going to infect cells that have CD4 plus one of the co-receptors, so like CCR5 or CXCR4. And so what you've done here with this VSVG, this vesicular stomatovirus, glycoprotein coat that you've put on, is you've given this virus the ability to pretty much get into almost any cell. So you have to be really careful when you work with this. What you do, so you don't kill yourself, is you take this P&L 4.3 modified plasmid HIV sequence. You've taken out some of the pathogenic virulence factors so that it is not quite as virulent. You've also taken away its ability to replicate. So even if it gets into a cell, it's not going to then integrate and have the machinery to come back out with the thousands, millions of little virions. And this is a tool that I use actually to manipulate the genetic sequence of stem cells. Okay. So I use P&L 4.3 with a VSVG envelope to actually rewrite the sequence of stem cells. And what they're doing here in the lab, using this tool to rewrite the genetic code of the human blood fluke, which is a wonderful tool because now you could say, you know what, I want, Schistosome to express gene X. And all you do is you clone that into this construct, and you can now put that gene into the cells of the Schistosome. So it's fantastic technology.
Very cool.
But not quite as frightening as when I read the title.
Yeah.
I thought I now had to worry.
I thought the same thing initially. I wasn't thinking of vectors. Yeah.
Yeah.
But of course, HIV would not be able to get into schistosomes because they don't produce CD4 in the co-receptors, right? So exactly. We should have thought, but in the current climate, you just worry.
I am very worried.
You do.
That.
So as Daniel said, they make a pseudotype with a VSV glycoprotein. They infect schistosome. What form of the schistosome do they use, Dickson? Schistosomula? What is that?
This is the immature stage of the parasite before it matures to an adult. After it enters, they collect it from lungs, as I recall.
Yeah. So they use schistosomula, and actually worms, they add virus. They can see its binding. They can see it's binding to the surface. They can detect reverse transcription in the worm by detection of DNA, by PCR. And they can see it go into the nucleus.
Exactly.
And one thing I was really very concerned about initially, Daniel, was that they threw in some reverse transcriptase inhibitors, AZT and nevirapine, and it didn't block reverse transcription. So I said, oh, that's not good. But they argue later that maybe the drugs never even got into the worm, right?
Right.
Yeah, that was my take. Is that so? They do the same controls. Actually, it's very nice. I do very similar work. They do the same controls where you heat and activate your virus to make sure that you're actually doing something versus a heat inactivated, which won't do anything. But then I also often use nevirapine, which is basically a reverse transcriptase inhibitor that penetrates human cells well. But I suspect, as they're suggesting, you're just not getting good penetration into these cells.
Then the next thing they do is they say, okay, where is this HIV integrated?
Right.
Right. And now you can do this on a massive scale. You can do whole genome sequencing. And what they do is they amplify the proviruses, which are the integrated HIV genomes, using a primer which is on transposons, which are known to be throughout the genome of the organism. They amplify, they sequence it. They identify 8,000 integrations. And they seem to be in all 8 chromosomes of the parasite.
Plus the sex chromosome. I mean, it was in all of them. That's right.
They also find it in mitochondrial DNA. What do you make of that, Daniel?
Wow.
It's amazing. Basically, it's going everywhere.
Because, this is going into mitochondria besides the nucleus, which is strange to me.
Right.
It's really interesting, and I have to say, I've never looked at that when I do like my human stem cell stuff, but it would be actually interesting to ask that question is, when I'm messing around with my stem cells and I'm, rewriting the nuclear code, what am I doing to the mitochondria in these stem cells? Because we use this technology to do gene therapy for things like sickle cell anemia and thalassemia, several of the other human genetic diseases, and never really focused much on, we'll say, off-target impacts on the mitochondria.
Daniel, has anyone looked in, not just you, but anyone looked in human mitochondria in infected people to see if there's integration?
Not that I'm aware of, actually. It's a very interesting topic.
So then they say, where are these integrations? Exons have 4% of them. So exons, of course, are the coding regions of proteins. Introns, 35%, and 62% in non-coding regions. That's where the integrations are taking place. I don't know why that you would have that kind of bias. Maybe because it doesn't disrupt. They're selecting for living worms, I guess, Daniel. So.
Yeah,
you're selecting for integrations that don't kill the worm, right? I think that's possibly the selection there. And then they do whole genome sequencing, and they say there are about 2 integrations per nucleus.
And a lot of that you can manipulate, in all honesty, by just how much virus you expose.
Right. Right.
So that you put in. I do a lot of where we'll use the ultracentrifuge to create these incredibly concentrated viral preparations, and you use a microliter and get a tremendous amount. So a lot of how many integrations you get, you can actually titrate based upon the exposure of virus, the viral inoculum. So,
of course, now this is useful for manipulating the schistosome, as Dickson said. And one of the things they want to do is use CRISPR, so they can disrupt genes or modify them, and they can do it with HIV now. So will we see an explosion of research? Well, this is 2016, Dickson.
It is.
So have people used this to modify?
That's a very good question.
I feel like there was I feel like there might have been a Trachanella paper that I saw in the last year or so where they were starting to use this technique for a few other. Because it's a powerful technique. The mitochondrial thing is interesting, and I don't know if I know as much about because whether the mitochondria lacks a certain membrane around it, or whether it's mitochondria are different in schistosomuli than they would be in a human cell. But that's sort of an issue because you're trying to manipulate the nuclear DNA. Yeah. It'd be interesting if there's a way to do something with the mitochondrial targeting.
Well, Dickson, Trachanella enters the modern world.
Stop it. No, kicking and screaming, that can't possibly be right. I'm just curious as to how the lentivirus might get into the Trachanella worm, though, because they've got this impervious outer cuticle, which the schistosomes and the tapeworms do not. They're basically exposed cells that would be susceptible to this technology, but unless you injected them directly into the cytoplasm of an adult female, let's say, that had eggs in it, that you could then allow the lentiviruses to integrate into the eggs themselves. I don't know. I don't understand how Trichinella could function in this way. But I'd like to read the papers and check it out.
Yeah, I mean, it's interesting, right, that what you can do with this technology is you can put whatever coat you want. Like I've actually put like feline viral coats on and other types of, outer coats to change the tropism. So one of the things you could do with potentially with Trichinella is, one, look for a stage where there's the ability to expose it and get cellular entry. And the other would be to specifically use a Trichinella-specific viral glycoprotein coat to pseudotype it and get it in there.
Yeah, kicking and screaming into the modern world. You're absolutely right.
Dickson, you said that you were not a virologist, but
you're right.
You're an honorary virologist because
honorary.
You've been on TWiP for 12 years, isn't that
This
right?
is true. This is true. And a lot of it has just sunk in. There's no question about it. So maybe that's why I found this article so appealing.
Dickson, how many genome types are there?
7.
Very good. Dickson, do you have a hero?
Do I get my candy now?
Yeah, you get your candy.
I do have a hero, and it's a hero which we've never talked about before on this show, but it's one which we should really talk about now, especially in these times. And the hero, in my estimation, and I think in yours also, Vincent, is none other than our own Daniel Griffin, who,
that's right,
through his continued vigilance at so many different levels of co-founding the Parasites Without Borders group, of interfacing with an outbreak of a very potentially dangerous disease, and at the same time finding time in his life for not only his family, but for sailing, for talking to us on TWiP, for coming on TWiV at various times. I don't know anybody else that could have actually done this. I haven't seen your red S and your cape yet, but I'm sure that you've got it hidden away someplace in a telephone booth outside. So that's what you change into the moment you go to work. I have deep admiration for what you're doing, Daniel, and I want you to know that personally, you are one of my all-time heroes. And I realize that you were a former student of mine. So I kneel at your feet and say, thank God you were listening to something that was being said while you were in class. No, I didn't mean to say it that way. Because you have an incredible latitude for including knowledge that most other people would just simply avoid and go on to something more understandable. You attack the most difficult subjects and come up with unique answers that have deep meaning. So for that, I have deep respect for your intellect and also for your friendship. So I'm offering you my choice of you as our hero this week, and I hope you'll accept that.
That is very, very kind. I'm glad this is a podcast. You can't see that I'm blushing.
No, but we can say flattered. That you're blushing. We can actually say
okay, yeah, I can't say it now. No, that was very, very kind, very generous.
Yeah.
It's heartfelt.
Very much deserved, for sure.
Absolutely.
All right. Now that you're a hero, you have to still give us a new case study, Daniel.
Yes, I have many. We will pick one for today. I enjoyed when I saw this individual. So the new case for today: a 60-year-old woman comes in with her husband, and the husband is apologizing, saying, "I'm so sorry that they're wasting my time." He thinks they should have just canceled the appointment. I'm like, no, go ahead. Tell me the story. And so I turned to the wife, and the wife reports that she grew up in Lima, Peru. So, of course, I'm already interested. I'm glad they came in. And we talk a bit about Lima, Peru. And she reports that she's now having an issue that she had when she was a child in Peru. She feels like it might be the same thing. And she says that as a child, she would get this itchiness around her anus, and that her mother would have her eat semillas de calabaza, so pumpkin seeds.
Huh.
And so she's having this problem, and reports that about every 2 to 3 weeks she'll wake up with severe itching. And when this last happened, she woke in the middle of the night. She was bothered enough that she went and she put a Q-tip just into the rectum and then extracted something of which she has a picture. So she saw her primary medical doctor first, who went ahead and ordered a stool ovum parasite, as well as a stool culture. And this all came back negative. So the PMD, primary medical doctor, was not sure what it was and referred her to me. Okay. Just a little more, actually not much more, which is that her past medical history is negative, no prior surgeries, she's not allergic to anything, no significant family history. She doesn't report being on any medications. She does do a bit of travel. And where does she travel to? She frequently travels to Uganda, where her husband,
Get out of here.
and where she and her husband work with a not-for-profit that does renewable energy products
I'll be
and
darned.
projects and puts in deep wells. So very, very interesting. We got a chance to speak a little bit about the time I spent in Lima and the time I spent in Uganda. And hopefully at some point they may help us out with some of the work in Baduda. But then, as mentioned, she asked me if I want to see the photograph that she took, which I do look at.
Absolutely.
And it isn't just a photograph. It's actually like a little video. And I don't think I even have to tell people at this point. I think I've given them enough. I think they're going to be able to guess what it is that we're going to see on the video.
In Uganda again, that's quite interesting.
Yeah, I thought it was really interesting when she and her husband reported that that's where they go to do this work.
Now, she is HIV negative, I presume?
She is HIV negative, otherwise healthy. And the primary medical doctor did, full workup, did blood work and chemistries, probably more than we needed to get this diagnosis. I think our emailers will let us know. But yeah, otherwise completely negative. No toxic habits, as Vincent likes to ask. Normal dietary habits. There's no pets in the house.
And she's living in the area, in the New York area.
She's living in the New York area.
Okay.
I like to say in a private residence with a concrete floor. No,
I was going to ask. Yes, that's why I asked that in the
Yeah, no, she's actually living in a nice home. Her and her husband are well off financially. He's done quite well, and so.
All right,
husband. Wait a minute. The husband has.
Oh yes, Dickson, you have a question.
I do, of course. And what about the husband? Did we say what happened to him?
Oh, yes. What about the husband?
So he's not reporting any problems.
None.
No.
Do they have any children or grandchildren?
Oh, that's an excellent question.
I see.
You're not going to answer.
Yeah, children, grandchildren, those are excellent questions.
Okay. I'm sure they have excellent answers also.
All right.
Good.
Let me see here. Let me read two quick emails, okay?
Please.
One is from Ryan. And they're both influenced by SARS-CoV-2, as you might guess. I've read news articles that hydroxychloroquine was originally used for malaria and lupus prior to SARS-CoV-2 testing. When anti-malaria drugs become non-effective, what will be the alternative treatments? When lupus treatments are not effective or are no longer accessed with hydroxychloroquine, what will be the alternatives going to be? Where do I look? Is there a phage that can attack only Plasmodium as a way to bypass anti-malarial resistance?
Wouldn't that be nice?
Well, phages are only on bacteria.
That's right.
Dickson, are there viruses of Plasmodium? For sure. Right?
There are. Of course there are. But whether they have any relevance to their pathogenesis or whether they can be used as a weapon against the parasite, we have no idea yet.
So I guess
it's a double-stranded RNA viruses, by the way.
Right. I guess Ryan's concerned about not having access to these drugs because of SARS-CoV-2. Is that the correct interpretation?
Yeah, exactly.
There's also the interesting issue he's bringing up, right? If malaria can become resistant to hydroxychloroquine,
right?
What will be the alternative? Like, if it turns out that these drugs do have a role and are proven to be effective in treating COVID-19, will resistance develop?
Right.
So interesting questions.
Good questions. Gesundheit.
So,
and it is interesting, right? The crossover between COVID-19 and anti-malaria.
Crazy.
Speaking of crossover, so you may have seen articles saying that ivermectin inhibits SARS-CoV-2 reproduction in cell culture. So we did a TWiP on Tuesday, and I had my wife, Doris Cully, who's an ivermectin expert, come on the show and explain that the levels they need, the micromolar levels they needed for inhibition of the virus in cell culture are not reached by the current approved FDA. The levels you need for river blindness, for example, are micromolar.
Right.
And she said you would have to do a toxicity study first before you could, because you'd have to ask the FDA for dosage trial because we don't have enough. They said that the amount you take now for scabies or river blindness is not sufficient to inhibit the virus. So she was quite interesting. Only time I could get her on TWiP when her drug came up. Christine writes, "Good day, professors. Strange days." She asked me, Dickson, why do you guys talk about the weather at the beginning all the time? Strange days we are in right now. Over the last week, Canada has been locking down to try and prevent spread of the virus. I don't disagree with it at all, but as a small business owner and no government assistance yet, we're scared and worried. Our sales have dropped 80%, and we're only 6 months old, so this has the potential to be devastating. And we don't know what to expect for the future. Experiencing something like this is almost surreal. I have a quick question. I hope to get this in before your next episode. My boyfriend read an article saying that the virus death rate is probably closer to 1% because a lot of people have caught the virus, but because they're healthy, they just think it's a flu and aren't being counted in the virus totals. But if they were, it would drop the death percentage. What are your thoughts on that? I would also like to thank you and everyone come on the show to provide us listeners with fact-based information. I wish the whole world was listening to you and not the media. I believe it would be a much calmer and informed situation if they did. Instead, we have people panic shopping, killing bats in national parks, and avoiding the Asian community because, being Asian or a bat must mean they automatically have the virus. I mean, I feel like we are devolving as a species.
Yeah, we are.
I've also attached a little cartoon I came across that it was kind of cute. Hope you like it. Wishing you and everyone listening the best, and don't forget to wash your hands.
Here.
Cute cartoon.
Yeah.
I think we are devolving as a species sometimes. I agree with you. So, Daniel, the death rate, do you think because we're not diagnosing a lot, the death rate is lower than we think it is?
So my hope is we've resolved this issue in the next few weeks with the introduction of serology testing.
Right.
Where if we can test, 10,000 people, which I think we'll quickly accomplish in a matter of weeks. Actually, the assay that we're trying to get up and running for my organization, I'll say Pro Health, is going to have the ability to do 10,000 tests a day. And a lot of people are very curious, have I had this before? So my hope is in the next few weeks, we actually have numbers. We can look at a large population and say, who's had it? And then, Pro Health, we take care of about 3 million patients. So we can very quickly look at numbers. Okay, how many people have had it versus how many people died and get this number. And I do, we hope it's as low as possible. Actually, 1% seems to me like it might be reasonable.
All right. Very good. Looking forward to that. I did a TWiP today with a scientist in Germany who's a coronavirus guy. And he said he's worried that if you're looking for IgM, you're going to get cross-reactivity with other coronas because the IgMs tend to cross-react. And so he said, hopefully you have to get people later and after infection, so you're getting IgG. And I don't know what your assay is going to do, Daniel, but... Yeah,
that was a little bit of the delay in us getting up and running, is that we wanted to make sure that we had an assay that didn't cross-react with the common coronaviruses, which there's about 4 of them. And one of them is actually a beta coronavirus, right? So it's alpha, beta, gamma. There's different sort of little. And the HKU1 is beta coronavirus, and the genetic code is quite different, right? So there isn't a lot of T cell cross reactivity, but the morphology of the spike protein is similar enough that if you're not careful in what assay, you can get cross reactivity even at the IgG level. So we basically took a little extra time, and I think it was smart, so that we can have an assay that doesn't have that cross reactivity.
Good, very good. All right, that is TWiP 182. You can find it on any podcast player. The show notes are at microbe.tv/TWiP. If you like what we do, consider supporting us financially, microbe.tv/contribute. And your guesses and questions and comments, TWiP at microbe.tv. Daniel Griffin's at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, a pleasure.
Dickson Despommier, Trichinella.org, thelivingriver.org. Thank you, Dickson.
Good stuff. A lot of fun.
I'm Vincent Racaniello. You can find me at virology.blog. I want to thank Ronald Jenkins for the music and ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.